I'm gonna. All right, so I have the privilege of introducing today's speaker and would like to thank Andrew Beckerman for joining us from all the way across the Atlantic Ocean in the UK, where he is a professor in the Department of Animal and Plant Sciences, the University of Sheffield. He generally works on food webs and phenotypic plasticity. And the coolest part of this is he has used Daphnia and algae. So that's where some of my interest in this comes in, to explore experimentally key questions about inducible defenses, with a particular focus on links between evolutionary ecology, developmental biology, and evolutionary genetics. And with that, I'd like to hand the stage over to Andrew, and it's all yours. Thank you again for coming.
That's great. Thanks, Paul. Yeah, it's really, really nice to be here. And as we were talking before some of you came on, it's possibly the only positive out of the COVID situation is actually having been forced to give seminars this way and realizing that we can travel internationally very easily now through Zoom and Google Meet and all those things and do this kind of stuff. So it's really, really exciting to join you. I'm in Sheffield, which is about 2 hours due north of London in the UK, and just on the border with a national park called the Peak District National Park. And just outside the window over here in front of me is one of the big reservoirs just on the edge of the park, with a giant overflow, which is on the verge of flooding because we've had 3 days of rain and they named the storm Christoph, and it's very exciting outside. So we've got reams of COVID, reams of rain, and I'm going to talk to you about climate change and food webs today. So thanks for coming. A little bit background, again, as Paul said, I've worked with Daphnia, with food webs, and with parrots. The Daphnia work over the last 20 odd years has been focused on inducible defenses, particularly interested in the genetics that underpin why inducible defenses, or how inducible defenses are formed and what triggers them. And with algae, the way that they induce clumping behavior has been really interesting as well as a form of defense. And that's work that is linked both to the ecology and to biotechnology, because clumping helps harvesting. One of the motivating interests I have in looking at the Daphnia has been thinking about foraging biology and traits and how it structures consumer resource or predator-prey interactions. And that's what kept me interested and led me back to the work I've been doing recently on food webs, which I'm going to talk to you about today. And then I also work on parrots in the Caribbean. No funding at all from the film series at all, but it is a really nice, lovely place in the Southern Caribbean to work on the parrots and conservation. But as I said, the focus of today's talk is mostly going to be around the food webs and food web networks, and a lot of computer modeling that we're doing to try and connect together how population dynamics and the traits, and particularly behavioral traits around foraging, can help us understand how communities will respond to environmental change. So today I'm going to talk to you about the work that we're doing centered around 2 research projects that have been funded recently, one on the ecological impacts of multiple simultaneous stressors, and the second is focused on stability in those ecological networks and whether stability in populations and the population dynamics gives us a good indication of stability at higher ecological scales, like when we look at the stability of biodiversity and the stability of ecosystem function. In order to start this story and give you an idea about how we think about this, I want to start with some work that we did more than a decade ago on the origins of food web complexity. And I want to give you this little introduction as a way to give you the vocabulary to understand what we're doing with some of the models of the networks that we're working with now. So when you look at that food web on the left, which is possibly a picture that you're familiar with, it's got some structure to it. The nodes are species, and the lines that connect them represent species that eat each other. So the things at the top eat things below them, etc. One of the tricks in network biology is that you can take these fancy pictures that look like the ones on the left, and you can turn them into the really boring ones that look like on the right. And we call that the matrix representation, or topological representation of the food web. And what you can see on the top are the letters corresponding to the species in the food web. And then the same letters running down the left-hand side where we treat them as resources. And every place that you see one of these gray squares is a feeding link. So species B eats species A, species B, and species C. And you notice I said species B eats species B, so you're seeing cannibalism in there, and you're seeing species B also eat other species in A and C. And there's a lot of stuff that you can gather from a picture like this, and they're really, really helpful in understanding how food webs are structured and what we look for when they're disturbed. One of the most important things to note about food webs when we pull them from these really nice pictures into this matrix representation is that they are non-random. And on the right, you can see a random matrix. So this is where we just fill in the cells randomly, or on the left with a non-random. And the non-random picture you see here is really characteristic. There's a couple of things to note. You'll notice that most of the gray dots are in the upper right-hand corner of the picture. And you'll also notice that there are sequences of squares, and those squares represent the diets of the consumers. In fact, there's lots of things that you can measure using a topological framework like this. We can look at species richness, so that's the number of species that are in the food web. And we can also look at connectance, or what we call complexity, which is the proportion of the spaces that you can see there filled with gray dots. Okay, and that's the proportion of potential feeding links that are actually realized. You can also detect what we call diet breadth, which is the length of those vertical lines. So B has a diet breadth of 3, whereas the species G has many more things that it eats. And you can see that that has a pattern across that food web. We can also look at vulnerability. Vulnerability is what happens when you look across a row there. So you can look at species C is fed on by species B, G, and H, whereas species B is fed on by even more species, both B, E, F, G, and H. So some species are more vulnerable to predation and being consumed than others. The generalism is that diet breadth that you see there. So the length of the bars vertically is how generalist the species are. There's lots more that you can do. You can get aspects of trophic levels, average trophic chain length, compartments, nestedness, modularity, lots of stuff thrown around. And what we call, these are all structural characteristics of food webs. And they're very, very characteristic and non-random. Connectance is really important because connectance, it appears, can give us insight into all the other features of the food webs. And we know this a little bit from theory. In 2001, Rich Williams and Neil Martinez published a paper in Nature called Simple Rules Yield Complex Food Webs. And what they did was to build a simple model where the structure of food webs, that pattern set that we were looking at there, is a function of connectance, that complexity. It's a little bit more complicated than that. It's a probabilistic model, so it uses some interesting statistics and distributions. But basically, if you know connectance, then you can predict all of those other characteristics. And you can see in the graph on the right, so the x-axis is normalized error. That's how bad we are at predicting those other characteristics. And under a random food web, random structure, you get all of that variation. Another model, the cascade model, which basically says just throw things randomly into the upper right-hand corner, it gets better. But with the niche model, the error in predicting what real food webs look like and that structure is really narrow, and it's really, really good. However, you have to know what connectance is. And to know what connectance is, you have to know what the food web looks like and who eats who. So just a little bit over a decade ago, we got really intrigued by that question of where does that connectance actually come from? Because if you know connectance, you know structure. If you know connectance, everything else emerges. But what if you don't know connectance? So what is the origin of connectance? There's a little bit of this potted history I want to share with you. Connectance is about prey choice. It's about the diet chosen by consumers, because that's what determines where those gray dots are. We know from classic ecological theory and lots of experiments over many decades that prey choice is not random. Some of you in some of your courses may have seen this picture in some of the books, the ecology books around optimal foraging. And the theory and the data suggest that animals feed on items that are not in proportion to their availability in the environment. What they do is to choose the items that are most energetically rich. They choose to maximize their energy intake, not to feed on the things that are most available. So you can see the crab there on the left, just as one example. The distribution of mussels in the environment is along the x-axis, and those are the little bars. But the energy gain that the crabs get is actually shifted to the right. So the mussels that are of length 20 are the most profitable items, and that's what they actually choose. You see this time and time again in organisms feeding on their prey. They choose the most profitable items, not the most abundant items. Now, this is embedded and core to optimal foraging theory. And that's the idea that you can predict the items that organisms eat based on the energy value, the time it takes to handle them, and the rate at which you encounter those items. Basically, what it's dealing with is translating calories and this idea of consuming items that you encounter into a choice that you make of what to eat. There's a whole lot of theory around this, really classic theory that goes back a very, very long time, to even a little bit before even I was born. And it's embedded in this really nice book, Foraging Theory. If you ever want to get into it, it's one of the best and most lucid descriptions of how all of this stuff works. The simple idea is that you choose the foods that maximize the rate of energy intake. And there's a little bit of a model. Here's a little bit of a maths equation that goes in there. And the lambda, the E, and the H are those energy values, the encounter rates, and the handling times. Now, hopefully what you see that I've done here is to start talking to you about diet breadth, which underpins complexity, we think, in terms of traits of organisms. And I'm sure you're hearing a lot about traits in the courses that you're taking and the research that you might be doing. Traits are really important. So the idea is that optimal foraging predicts the diet breadth of a consumer. And if we know that for a whole bunch of consumers, then the complexity of a food web made up of all those consumers is the average diet breadth among all of them. And that was a big step that we made. And here's a little bit more maths if you wanted to go through it. The stuff on the left, the D, is the diet breadth of any individual species. Again, that's that tool we can use to predict what animals eat. And then on the right is how you move backwards from diet breadth, average diet breadth on the right-hand side, D over S, all the way back to connectins, this complexity of the food web. So this was our idea. Can foraging theory, based around these three simple traits, actually predict the complexity of food webs and thus the structure of those food webs? So what we did was built this little algorithm. You can check out the papers. They're back from 2006 and 2008 if you want. And it's based around that simple theory. And this is what we did. We said, okay, we've got a number of species. Let's say there's 20 species. We define for each of those species the energy value, the handling time, and the encounter rate. Okay. You do that for every species as a consumer, potentially feeding on the other species as a resource. So we're trying to build that matrix that you see on the right. For each consumer, you go through this process of applying the theory, and you apply the ranking of profitability, and you ask which ones should they eat, and you stick those little dots into the grid to predict what complexity might look like. You build the matrix of links. Now, the awesome thing about this is that it worked. We were able to calculate connectins, and that's what we show in the 2006 paper. What we hadn't been able to do with that paper is actually show that it produced an understanding of the complexity and structure of those food webs to make it look like you see there on the right. So diet breadth predicts complexity, but what about the structure? How do we do that? It was another simple step. And it's the idea that these traits like energy value, which is really related to size, handling times, again, related to size, and encounter rates, also related to size. This idea of allometry, that traits scale with body size. There's lots of data out there that suggests that these traits do scale with body size, and we collected a lot of information on it and came up with a way to modify our algorithm so that we were choosing these parameters as a function of the body size of the individuals. Having done that, we were not only able to predict the connectins, but also predict to a very good degree, based on the traits, the structure of the food webs. Okay, so some of these look a lot better than others, definitely. But at the time, we produced a model that was the first to use traits to predict complexity and structure of the food webs. When you have very, very good data on body size, like this example here in a recent issue of Nature Climate Change, my colleague Owen O'Gorman and Owen Pecci, who did the other work with, you can actually use this model, this diet breath model, to capture between 60 and 82% of the feeding interactions across a range of environments and food web properties. So it's very good at predicting the complexity and structure. And it's all based on traits and foraging biology. So that's a little potted history of something that I need you to understand as we move into the next part of the talk. So foraging traits predict and body size predict food web complexity and structure. Okay, let's shift gears a moment and let's talk about threats that ecosystems face. And then we're going to come back to how we've been using that idea of traits to do some modeling to address our understanding of threats. I'm going to play a little game here. There are lots of threats to communities, anthropogenic sources of stress that organisms and environments are facing. We've got a bunch of them listed here: temperature change, fertilizer runoff, hunting, pollution, heavy metals, urbanization, pesticides, acidification. Why don't you just take a moment and have a look at that and think, if I were to ask you to categorize these, what might you do? So just have a look at those, maybe jot down some notes even, give you a minute. Would you put any of them into specific categories, and why would you put them into those different categories? Don't worry, I won't ask you to say anything. Just go ahead and do that process of thinking about categories that these fall into. So one thing you might notice is that some of them are distinctly associated with the killing or death of organisms. Hunting and fishing takes away numbers of individuals. You might look at some of them and say, sometimes things like pollution or heavy metals do that as well. But we're also now into a zone of life and environmental regulation where things like heavy metals and pollution are entering at what we call sublethal levels. So they're operating not directly on the numbers of individuals, but indirectly on them through physiology and life history. You might look at some of them and think, those are at a very different scale than the other ones. Deforestation, that's taking whole communities and impacting them, wiping out everything and leaving it bare ground if you're doing a clear felling. This is a game we played in trying to get this work funded, and we thought about it this way. So you can think, again, I've just noted there, these are affecting births and death rates because they're changing numbers. One of the things that we did was to think about these categories like this. On the left, you can see things that are impacting through physiology on the sublethal level, but also through birth and death, right? Because we know that things like pesticides, acidification, heavy metals, even temperature, there are lethal limits to them. Things like fishing and hunting, they, in our heads, fall into this category of definitely impacting on the numbers directly. But we might also think that there is a risk factor associated, so there could be some physiology and life history going on there too. Things like fertilizer runoff, they're affecting whole trophic levels because the nutrients that are coming in are affecting all the plant resources, all the basal level of a food web. And then deforestation and urbanization, certainly at that big scale, but they might also be having effects down at other levels. But the simple idea here is that classifying the way we think about threats can help us understand how they impact on communities. Are they impacting on the numbers directly, on the traits, on large numbers of individuals, trophic levels, or on whole communities at once? When we classify the threats, we understand then that they enter communities at different ecological scales and therefore drive the impacts on the dynamics of species and the patterns of biodiversity in different ways. So we can think of this map here. There's our little grid, classified grid turned on its side. I'll just replace that with the words because it's a bit easier. And the ecological scales down the middle. And you can see we've got individuals and traits, population, trophic levels, the community, and possibly up to the ecosystem. And we can draw some arrows, physiology affecting then most directly on that individual subpopulation level, the birth and death at the population level, productivity at the trophic level, and then habitat at the community. But there are some other ways that they can have impact, but those dominant effects, I think they seem to make sense. What's really cool, again, is that the way those stressors impact systems and the way they enter communities then drives patterns of biomass, dynamics, stability, biodiversity, and ecosystem function, the things that we're most interested in understanding as responses to threats and climate change. So what we're hoping to be able to do is to take that idea where we think about traits and populations and communities and translate it into the ability to make predictions about what happens when multiple simultaneous threats impact on a community. Can we map the dynamics and the structure of the community through time in this framework? So threats enter communities at multiple ecological scales. They generate consequences at multiple ecological scales. And these are the things we measure in order to understand and assess the impacts of climate change and environmental change. And therefore, these are the things we hope to be able to predict. So, stage 3 of the talk. Let's step back to the foraging traits because I want to introduce one more thing about what that foraging biology means to our understanding of how to model ecosystems and communities. And the idea I want to introduce to you, which comes straight from those same traits and the same foraging biology that I talked about, is called rewiring. Rewiring centers around foraging biology, and we know that foraging biology predicts complexity and structure. What's interesting when you think about these stressors impacting on communities is that they're causing extinctions, or at least substantial lowering of the abundance of certain species. So species abundance are going down, the relative abundance of them are changing, and sometimes they're going extinct. This generates a response in other species. Species that eat something and then lose that item aren't necessarily going to sit around and go, oh, nothing left here in the fridge. They're going to go to the supermarket. They're going to look around and they're probably going to make a decision to try and eat something else unless they're an obligate specialist. Okay? It's going to generate responses in other species as well. Some of them are going to be released from predation pressure because of the way these diets might change and the changes in abundance happen. And it's going to change a response, it's going to generate a response at community level as well. The distribution of traits are going to change, and we know that that has impact on stability and dynamics. So let's just understand a little bit more about what rewiring means. Here's a little graphic. You can imagine we've got a little food web here with connections. Imagine this species gets hit by something really hard and goes extinct. That means the links between the species above it, the ones that eat it, have disappeared. And you'll notice that there's a sad species in the middle that no longer has any resources. And in classic thinking, that means that that species might go extinct as well. So we could get rid of that, leading to a secondary extinction. However, it's very possible that because of foraging theory and thinking about generalist species, that it could choose to eat other things. It could change its diet. And that's the simple idea. Organisms are active foragers. The loss of items from a diet can induce a reassessment of the profitability of the range of other items available for it to eat with respect to these traits that we talked about that underpin foraging biology. So diets can change after extinctions events. That's pretty cool. So we've known this for a while, and people have worked with these topological maps of food webs, these structural food web models that I've been showing you, where you've got the dots in the locations of what things eat. So we look at secondary extinctions. And one of my students, Aaron Theory, has done this, and then Phil Stanishenko as well, looking at the way in which these adaptive behaviors can increase the robustness of communities to the types of responses to ecosystem threats like extinction that might happen. Basically, foraging biology generates more flexibility, which allows ecosystems not to fall apart as quickly in the face of stressors. So let's put it all together now. A few years ago, there have been some major advances in modeling population dynamics of multi-species communities. There's a model known as the bioenergetic food web model, or the BEFW for short. And this model is based around simulating biomass dynamics and centers on the metabolism, the growth rate, and the foraging traits of species related to body size. The foraging traits in the bioenergetic model are exactly the same foraging traits that we use in the model to predict the complexity and structure of food webs. So all of a sudden, we've got a model about population dynamics that can be connected to a model that allows us to monitor and manage rewiring. We can allow populations to change through time, and we can follow the structure through time as well. So we can evaluate dynamics, diversity, and structure through time with this model that allows us to simulate the dynamics of species and how the network changes with rewiring. We can monitor stability, diversity change, and robustness to extinction with and without this rewiring. It's a very exciting situation to be in because this means that we can move towards making predictions and trying to make some generalizations about how the communities that we rely on in terms for food, for ecosystem services and ecosystem functions, how they respond to multiple simultaneous stressors. We've got a model that allows multiple entry points, a model that allows rewiring, and we can follow the dynamics of species, the dynamics of biodiversity, and the dynamics of ecosystem function. So how do we do this? We have this beautiful model that allows us to take all of these things into account. And what I want you to imagine is if we use this model in an experiment. I think a lot of you have learned about how to do experiments or do them yourselves or are doing them. It's great. And I'm going to present to you a factorial design where we manipulate temperature, the presence of copper, and the presence of nitrogen as a stimulant to the community, a fertilizer to the community. These are different types of stressors. They cover the classes that we've talked about, that classification, physiology, productivity, birth and death. And we have a factorial design, and then we're going to measure stuff at the population, community, and ecosystem level. And I'm going to do this for lots of different communities of different sizes, so we can vary the species richness. We can start with different structures, and we're going to plug it into this model. What we might measure are the abundances of a bunch of different species. You can see that in the yellow, so that's the population ecological scale. And we can then measure a bunch of stuff at the community level, like the biodiversity or species richness, this complexity metric, patterns of generalism, the distribution of body sizes, the robustness to extinction. And then at the ecosystem level, we can measure total biomass, the flux of energy, patterns of productivity, or even total ecosystem respiration rates. We can measure all of these things and make predictions about them with the model that we built. Now, what we do is no different than what you would do in a field experiment. We generate treatments, so variation in temperature, copper, and nitrogen, and then we can analyze the outputs of the model using statistics. We can gain estimates of the main effects of two-way interactions, the T by C, the T by N, and the C by N are temperature by copper. So the effective temperature depends on copper and vice versa. And then even the three-way interaction. And with some clever statistic stuff, we can use standardized effect sizes. So we can estimate these effects so that the scale at which you measure stuff doesn't matter anymore because population, community, and ecosystem scales are different. Basically, it standardizes the effect we measure and the size and impact of the different stressors. And it's with this approach that we can evaluate what we call predictability and generality. Now, predictability is a quite simple idea. When the different stressors add up, that's more predictable than if they are synergistic or antagonistic. And we detect synergy or antagonism when there are statistical interactions, when there's context dependence. When the effect of temperature on species abundance varies with copper, that's harder to predict than when the effect of temperature and copper just add up. So predictability is about whether or not the effect sizes are strongest when there are interactions or around the interactions rather than in the additive effects, which are just the single letters there. Generality is defined this way. It's when we see patterns in the distribution of those effect sizes. Are there clusters of effects among populations and or by threats? Sorry, among scales and by threats. Let's look at an example to see how that might work. So here's a picture, very stylized picture. It will never come out like this, but let's look at it this way so we can understand predictability and generality a bit better. What you can see here is that the population dynamics are way more predictable than the community structure and much more predictable than ecosystem function. And that's because all of the impacts in this stylized example at the population level are additive, whereas at the community and ecosystem level, it's all contingent, context dependent. You have to know exactly where you are with respect, particularly at the ecosystem level, to the levels of 3 different stressors to know what's going on. What do we see in terms of generality? Well, we can see that there are some patterns here, that populations are additive, communities are pairwise, and the ecosystem has this 3-way. There's another pattern in here as well. You can see that there are specific populations and specific traits and characteristics at the community and ecosystem level that are being impacted in the most severe way. So the effect of temperature, copper, and nitrogen on species 1 is the strongest compared to the other species. So species 1 is suffering, species richness is suffering, and biomass is suffering, where the other ones aren't suffering as much. So there's another pattern that you can distill there. Here's a more realistic pattern that we might see. It's a mess, okay? But if you look closely here, and I've highlighted the upper left again, you can see that predictability is on average lower compared to the other picture we saw, but the population level has more additive effects that are coming out than do the other ecological scales. Now, there's one other pattern in here as well, and I wonder if you guys can look carefully at the red squares. If you look at the red squares, they're associated with the same thing in every column. There's something about the columns they're in that's common. Can everyone see what it is? The letter T is common to all the red squares. T in the first one, 4th column over, T by C, you've got red. T by N, you've got lots of red. And T by C by N, you've got red. That means the strongest effect sizes we're detecting, so these negative impacts on species, community, and ecosystem traits, are all associated with the letter T, which is temperature. So this is an example where we might see a pattern that suggests that one stressor is a threat multiplier. And that's what a lot of people think about when they talk about temperature and climate change. It's not necessarily horrible on its own, it's a threat multiplier, and it's always associated with generating bad things when other things are going wrong as well. So this is the kind of stuff that we can do with the model. There's the picture of the generality. So what I want to do to finish up and give plenty of time for some questions that you might have is go through 3 examples, a little bit of where we are with trying to apply this model. hopefully give you some insights into the fact that it is working and it's not just these cartoons that we can build. And the first one I want to talk to you about is where we've looked at the frequency with which rewiring occurs and its impact on the robustness to extinction. This is work that my former postdoc, Benno Simmons, did, and it's based on this simple graph that I showed you before, where the robustness to extinction increases with this rewiring mechanism that we allow in the food webs. Remember that rewiring mechanism is a way to say, if a species loses something that it eats, it reassesses its diet and can change the links it has to other species. So the structure of the network can change, it's adaptive and flexible, and that allows the community to be more robust to extinction and secondary extinction cascades. However, most of the work that's been done in the past, both with topologies and with population dynamics, has focused on what happens if you allow this rewiring process to happen when the extinction happens. Now, we thought that was a little bit weird. Why would you only let this flexibility happen when there's a loss of a species? Doesn't it happen all the time? Benno did a lot of work and continues to do a lot of work with pollinator networks. And it's in that space where we have learned a lot about understanding rewiring and reconnecting and distributions of connections in networks. Because you can imagine in the summertime, you've got a bee, it goes and you see it feeding and pollinating one flower. The next day, it's using a different species. The following day, it's using a different species. And then it comes back to the first species you saw. Over the course of a week, the links being created are changing. Rewiring and reconnecting and making decisions about what consumers feed on or pollinate happens all the time. So Benno's idea, and one of the things that we've been working on, is allowing this rewiring to happen consistently in food webs. And the hypothesis that we've got is that with continuous rewiring, extinction can be averted. For some species, the time to extinction can be delayed, and for other species, this can dampen the variability in their dynamics. So Benno did a big experiment working in the computer with a bunch of species in a food web network, and we played the game of taking individual species out, doing a primary extinction when the rewiring was continuous versus when it was not. And we found exactly what we might expect to happen with that rewiring. You can see in the lower panel here, the V16 panel that I've highlighted, that the extinction has been averted when we allow rewiring to be consistently running. So you've got extinction rewiring in blue, and you can see the species goes extinct, but with continuous rewiring, this species does not go extinct. You can see in this panel that extinction can also be delayed. So the time it takes for the events to occur is extended. You can see again, the blue drops off at time 4 and the red at time 6. Finally, all the top panels have much lower variability in their dynamics under that extinction event. It's a little bit hard to see in there, but the reds are all underneath and the bandwidth of them is much narrower than the green ones, which are the extinction only rewiring events. So, and this is just a picture of that time to extinction again, with the continuous being much longer tailed than the the extinction only rewiring event. So it's this experiment that we've done with the computer and with this model that tells us that indeed the way robustness to extinction and response to threats increases is by averting, delaying, or damping the impacts that threats are having on the ecosystem. A second thing that we're doing is pursuing directly this question about how multiple stressors impact on communities. And we're doing that with and without rewiring again. So does allowing rewiring increase the persistence of species? Got some indication that it does from Benno's work. And does rewiring alter the predictability and generality? But this is under now multiple simultaneous threats to ecosystems. My postdoc Eva Delmas is working on this right now, and we're starting from a baseline of work that was done by Binzer et al. In 2016. What they did was to use that model that I've talked about, the bioenergetic model, without rewiring, and they simulated changes in eutrophication and temperature, and they measured the persistence of species in the food web. What we've done is redone that, and we're getting the same answers, which is good. And we're also running it right now. It's being run actually on a computer in Montreal with rewiring. And our objective here is to evaluate that grid that I showed you. Does persistence go up? Do these metrics change? But looking at multiple metrics across the ecological scales. So abundance patterns, species diversity, body mass distributions, and things like energy flux at the ecosystem level. Are there impacts of temperature and eutrophication that are different when we, and is the distribution of those impacts different when we allow rewiring to happen? It's a very robust way, this effect size grid, to make comparisons because they're all standardized effects, and we can look at those patterns and make strong inference about what's changing as we alter the presence and absence of rewiring and the magnitude of these different stressors. Ultimately, what we'll be doing with this is increasing it to 3 stressors and even 4 stressors, and looking again at that grid of impacts. Now, one of the other things that we're doing, and what I just mentioned there, is that you work on this grid of stressors. However, we know, and I think everyone would agree with me, I hope, that stress is not a discrete event. Sometimes it feels like it is, but things like temperature, things like pesticides, pollution, it's changing all the time, and it's generating gradients of impact and threat to ecosystems. One way to think about this is to look at the classic experimental design that people have done with the models, but also with experiments. And that is to take a particular community and stick it into, say, 3 different temperatures. You can see a temperature 0, 15, and 30 degrees. You start with the green food web in the community, you stick it into these 3 temperatures, you wait a little while, and boom, you've got a different structure, different species have survived, different abundances, etc. Now, this is really valuable because this kind of discrete event does happen. There are shock extreme events, temperatures go up, temperatures go down, got starting conditions. However, what's a little bit more realistic is that temperatures change through time. So you might have a community at 0 degrees in the middle of winter, moving into summer, moving into the height of summer, and then moving back into spring and back into autumn and then back into winter. So there are seasonal changes going on. And there are also changes that we know from all the predictions and all the reality about temperature change and other forms of climate change and environmental change. There are mean changes, changes in variance, and cycles in all of those stressors that we're interested in, threats that we're interested in. So one of the things that we're trying to do is understand how the predictions vary, particularly around stability of communities when you present a sequence of change rather than these discrete events. And here's a little bit of a teaser on some of the work that we've done. You can look on the left-hand side here, the y-axis is mean stability of the community. Okay, so this is looking at how variable the population dynamics are through time. And you can see that with a discrete model, the one at the top, where you stick everything into the different temperatures, we have quite high levels of stability. But when we allow this sequence to occur, stability is much reduced. Very big impact of changing the assumptions about how we design the experiments, and this matters both for empirical work and for the theoretical work that we're doing. So, wrapping it all up, foraging biology predicts food web complexity, structure, and underpins the dynamics of ecosystems. In fact, a set of very simple traits, energy value, handling times, encounter rates, and body size, seem to structure communities and give rise to the dynamics that we see. When we combine that with metabolism and growth rates, we get this beautiful picture and tool that allows us to work on understanding how threats impact ecosystems. And we played a little game in the middle of the talk about classifying threats. This helps us understand how to model the effects of multiple simultaneous stressors and make predictions and capture the understanding that the outcomes of different stressors are going to manifest at different ecological scales because their impacts are hitting at different ecological scales as well. And finally, predicting and generalizing the effects of multiple simultaneous stressors, I think it is possible. I think we've got a good rubric with which to do this, and we've got a model that captures a lot of really good biological realism in complex communities. And hopefully, maybe I can come back in a year or two time and give you some of those predictions and how they match up to some of the data that we have from fisheries, freshwater ecosystems, and avian communities as well throughout Europe. And with that, thank you very much for taking the time to listen to me this afternoon for you and this evening for me.
Thank you very much, Dr. Beckerman. I will say, first of all, that you made that incredibly, all of your modeling very straightforward to someone who does not have a background in ecology. So thank you for breaking that down so well. I know that it is not as straightforward as you made it seem, but you broke it down and it was very understandable. But we will open up for questions. So usually we have people write in the chat, but I also encourage anyone who does want to just unmute yourself and ask any questions you may have for Dr. Beckerman. So we will open up for questions.
Excellent.
I was wondering if you could explain a little bit more for the matrix version of the food web, why all the interactions are in the upper right corner of it.
Excellent. Yeah, that's a very good question. The biology behind that is that the ordering of that picture is by smallest to largest species. And you get the upper triangular picture typically in real food webs because small things don't typically eat big things. So you get small things eating small things and then the big things eating the bigger things. And that's why also with big things, they have broader diets as well. They have that bigger choice. Now, I'll just let you know, one of the things that's fascinated me about that is that it's not true for some organisms, particularly herbivores like caterpillars, things like that, which look like they feed on trees or large plants, things that are bigger than themselves, but also parasites, which are one of the most diverse and abundant groups of organisms on the planet, and they certainly don't consume things that are smaller than them. So there's lots of deviation from that pattern that comes when you start thinking about things other than just predators. Hopefully that's answered the question.
Yeah, that's really interesting. Thanks.
There we go.
Any other questions for Dr. Beckerman?
Hi, Tom Hutchinson. I would ask one question. First of all, Andrew, I thought that was a really excellent talk. I very much enjoyed it. It's sort of memory lane for me. I did my PhD at Sheffield many years ago in plant ecology.
Excellent.
In the days of Phil Grime and Harold Woolhouse.
Excellent.
So I think this is are you speaking from Sheffield? I haven't gleaned it. Are you speaking directly from Sheffield? That's amazing.
I am. I'm just on the outskirts of Sheffield near Damflask Reservoir near Bradfield, High Bradfield.
Okay. Well, one of your earliest slides, I think the simplest one was that the prey, they're going after the prey which has the most energetic benefit. Right. Is that not just simply that they go, I mean, it's also that means they're generally going after the bigger prey.
It can be that the bigger ones are more energetically valuable. The rules that make up the theory are that profitability is defined by energy value divided by the handling time of the prey.
Okay.
So there's two traits that govern that profitability. And then there's an encounter rate bit in there as well, which determines what you take as the first, what you take as subsequent items, because that installs additional costs to the organism.
Okay, so how would your tent caterpillar example from right at the end, or the caterpillars eating trees or leaves, or how does that fit into it?
Yeah, so one of the things I didn't tell you, and now I can be really open and honest because it's a great question, Tom, is the modeling, all the work that we've done is focused on predator-prey interactions. And these models work best in aquatic systems where typically the big things eat the small things and organisms are engulfing. So the fish, the cladocerans, everything basically chews on individual items. There's a whole nother class of foraging biology called patch foraging. And it's patch foraging that captures the way in which things like caterpillars and things like parasites forage much better. And I've got a PhD student right now, and half of their project is designed to elevate this foraging biology question and predictions to make it more realistic for the half of the organismal biota that we have actually ignored.
Okay, thank you.
Yeah.
Thanks very much.
I will say, just as an aside for those of you who are interested in that, one of the big steps in doing that is recognizing that a caterpillar doesn't eat a tree. I think, as Tom pointed out, it eats a leaf. And actually, caterpillars are really specific about bits of leaves they eat, and they can sense where the high nitrogen parts are. So the challenge quite often for translating the way in which certain organisms eat into this item idea is recognizing what the items are. It's not the leaf, it's not the branch, it's not the tree. And we've done a little bit of work where we can look at simple food webs where herbivores are heavily involved, and you see very distinct changes when you shift the definition of the item, and it brings the world of those organisms like herbivores, terrestrial herbivores, right in line with what we see empirically and theoretically in aquatic organisms.
I have a question. Thanks, Andrew, for the great talk. That was really a really great link between everything ranging from population dynamics all the way up to food webs and complexity and connectance. And I'm just wondering, I get the sense that most of these food web models implicitly assume or explicitly assume a type 2 functional response. And I'm wondering if there are additional models that consider other types of functional response, either a type 3, like a sigmoidal shape that accounts for prey switching, or models that incorporate predator dependence. And I'm just thinking also in the context of rewiring, I can see rewiring not only changing which prey types you go for, but also changing the actual shape or the nature of those specific resource consumer interactions. So changing it from a hyperbolic to a sigmoidal, or a sigmoidal to a hyperbolic. And those things can have pretty dramatic impacts on stability and population dynamics. And presumably those would translate all the way up to impacts at the food web level. I'm just wondering if people have been working on that question, or if your group has thought about those questions.
Yep, it's a fantastic set of questions and ideas in there. And the whole lab group just presented at the British Ecological Society meeting in December. And I would say that 90% of the questions we got centered around exactly what you just brought up. And the short answer is the population dynamic side of the model, where there is a functional response that defaults primarily to the type 2, has a tuning parameter so that it can move, the Hill exponent can move around to allow variability there in what goes on. It is worth pointing out that the database that we started and the evidence that continues to accrue from people doing studies on functional responses is that by far the type 2 functional response dominates the world. So that's not to diminish the impact of these other functional responses, but it is part of the model data interplay that we pay attention to. The longer answer will come with more research, and we are working on it. And it centers around the idea that the foraging model, the rewiring model, isn't a prey switching model per se. It's a contingency model of diet choice, and it is derived from a type 2 functional response. In fact, the two things are essentially equivalent. But it doesn't do the same stuff that a type 3 switching behavior would do. And what we are, because of all the comments like yours that we've gotten, what we will eventually play around with is working with the tuning, the Hill exponent parameter in the dynamic system and the rewiring tool, and looking at how they interact with each other. Really, really interesting stuff, because it's trying to get your head around how prey switching is happening, which is generally working at relative abundance of prey items in a diet, versus the link distribution changing, which is that big thing about links disappearing and coming back in. And they're both really important processes. So awesome question, really interesting stuff, making us go back to another really old body of literature in Oaten and Murdoch with the prey switching functional responses as well. Thank you.
I would say we have time for 1 or 2 more questions if anyone does have any lasting questions. All right, well, we can call that a wrap then. Thank you again, Dr. Beckerman. We really appreciate you giving this lovely seminar. And thanks for all our audience members. So I guess you can go to bed now. It looks dark. I see the dark lights.
Nice. It's dinner time, not bedtime. It's only coming up on 6 o'clock. Yeah, I'm not that old that I have to go to bed at this hour.